Hi, my name is Autumn Morrow, and I designed this course in immunology a while back. I designed it for my university, and then recently thought, I would like to share this with a much broader audience. And that's mainly because I believe everyone should be able to have free access to education, and quality education nonetheless. So this first episode is going to focus on the immune system and how it develops, and then later episodes will dive more into B cell and T cell differentiation. So we'll begin with hematopoiesis, which is basically the production of blood cells. Here we have hematopoietic stem cell, and this hematopoietic model is considered classic because it descends in this hierarchical manner. But recent advances in technology have been showing that these immune cells can actually differentiate in any direction. So this traditional model is still used for the sake of convenience. But it's also kind of cool and helpful to know that these immune cells are doing some very neat stuff by differentiating in any direction. We'll begin with exploring the two major arms of the immunity that happens in the body, and that would be the innate immune system and the adaptive immune system. So the way the immune system responds to its environment determines whether it's going to be innate or adaptive. Innate is not specific to a pathogen, while adaptive immunity is specific. So the innate immune system, when we say not specific, that means that it responds to the chemical properties of infections rather than the pathogen itself, where adaptive immunity is specific to an antigen that comes from a pathogen. And if you recall, an antigen is just basically a peptide with a shorter amino acid sequence than a protein. So you can sum it up by saying that the immune responses are primarily divided by 1, how specific the response is, and 2, the duration of the response. Innate immunity is a lot more acute, where adaptive immunity is more long-term because of its ability to create these memory B cells and T cells. Now we can drill in a little bit more to adaptive immunity, comprising cellular and humoral immunity. As you can see, cellular immunity, as it's called, primarily involves T cells, and they can either differentiate into cytotoxic T cells or T helper cells with different effector functions. And on this side, we have humoral immunity, and that's where we're going to find the B cells that differentiate into these plasma cells capable of secreting immunoglobulins, also known as antibodies. It's always helpful to know where you came from and where you're going, so that's what this slide is about. We begin again with the hematopoietic stem cell that develops in the bone marrow, and after traveling through circulation, these become mature in the thymus, and that's where T cells got their name, actually, is from thymus. So after maturing in the thymus, the T cells travel through the body until they encounter one of these antigen-presenting cells that show them a pathogen, and that's when they become activated in the lymphoid tissues. Now let's begin studying the innate immune system. So here are some key players in the immune system that might be helpful to review, along with their main functions. So here you have an epithelial cell, and that can be any cell on the skin or in the lining of the mouth or your organs, including the gut. Here we have a mast cell that lives in your mucosa and connective tissues. They also play a big role in allergic diseases. So here you have the granulocytes, and those include neutrophils, which are phagocytic, and eosinophils and basophils that can respond to parasites but are also problematic in allergic diseases. Here you have a monocyte. These are attracted to infected tissues where they turn into macrophages. And here are the macrophages and the dendritic cells. And these are antigen-presenting cells that help to activate the T cell response. That's how they're able to serve a bridge between the innate and the adaptive immune systems. Lastly, we have a natural killer cell that can induce apoptosis of infected cells. And while natural killer cells are part of the innate immune system and are non-specific, we have T cells, and these are adaptive and specific to a pathogen. T cells also activate B cells, generate memory cells, and regulate both of the arms of the innate and the adaptive immune system. Now we'll focus a little bit on these antigen-presenting cells, also known as APCs. So here are the professional antigen-presenting cells. We have macrophages and dendritic cells from the innate immune system, and then B cells from the adaptive immune system. All of these can present antigens, hence their name, and activate the T cell response, and they can all serve as a bridge between the innate and the adaptive immune system that way. Antigen presentation happens with these things called major histocompatibility complexes. We have MHC-I and MHC-II. We also have B cell receptors that are surface immunoglobulins, and these can detect and ingest antigens through endocytosis. After the B cell becomes activated, the peptide is then displayed on the major histocompatibility complex 2. Try saying that fast twice. So here's another table for professional APC functions. Again, macrophages, dendritic cells, B cells, innate, and adaptive. Antigen presentation. Dendritic cells are the only cells that can present both MHC-I and MHC-II. We'll cover that a little bit in more detail later. Macrophages are highly phagocytic, while dendritic cells are actually the best at presenting antigens. B cells are also really good at presenting antigens, but they're not as good at phagocytosis. So MHC-I is actually present on all human cells, including epithelial cells. And this is really helpful for the immune system to determine whether a human cell is healthy or infected. In the case that it's infected, the cell will either undergo apoptosis or be flagged by the immune system for destruction. As you can see here, MHC-I activates the CD8 T cell response, and when those are activated, they turn into cytotoxic T cells that help to fight infection. MHC-II, on the other hand, is going to be found on just macrophages, dendritic cells, and B cells. MHC-II is really helpful for guiding the differentiation of naive CD4 cells into mature T helper cells with effector functions. Now, the only antigen-presenting cell that's capable of expressing both MHC-I and MHC-II for pathogens is the dendritic cell. And that's really important because it actually creates this co-stimulation between T cells that ends up helping and enhancing the differentiation process. We had talked about the MHC-I in the last slide, how it can display either an antigen from the cell showing that, hey, I'm a healthy human, or in the case of an infection, MHC-I can express an antigen from the pathogen that shows, hey, immune system, I'm infected, I need to be killed over here. And then that's how your body protects itself from infection. So for illustration purposes, I created this little guy, and this is going to represent an antigen-presenting cell. Most commonly, it's going to be a dendritic cell or macrophage, but I combined them both into one for sake of convenience in some of the future slides. Now this is where it gets exciting because we have the innate immune system meeting the adaptive immune system. So we talked about how an APC can ingest a pathogen and then display a part of the pathogen on its cellular surface. And that's so that other T cells can recognize it and then get activated. The way that they get activated is through these cytokines, these signaling molecules in the environment that are released by a variety of APCs. And that helps to guide and give the instructions to the naive T cell. And then the T cell can become one of any of these T helper cells with effector functions that match the pathogen it's intended to fight. So real quickly, there are 3 factors that affect differentiation. 1 would be the strength of the antigen, 2 would be the signaling environment composed of cytokines, and then 3 would be the interaction with antigen-presenting cells, the types of bonding and communication that occur between the two. Here are the different types of cytokines that might exist in the microenvironment, and depending on what composition you find will determine what the T cell ends up differentiating into. The naive T cell can differentiate into any of these T helper cells that have all of these different effector functions. Not only do they have different effector functions, they can also release cytokines of their own that end up furthering the immune response. Here's another table that helps to summarize the different T helper cell subtypes, and then what they're responding to, and what cytokines they produce in order to achieve which type of outcome. Now, I'm not going to spend a lot of time on this table, but it is a nice summary, and we will be diving into each one of these T helper cell reactions in later episodes. If you want to stay tuned, feel free again to follow my channel and stay posted that way. Now let's cover the antibody response in the adaptive immune system. We'll begin with covering some immunoglobulin functions, including opsonization, neutralization, and activation of complement. So antibody opsonization occurs after an antibody attaches to a surface antigen on a pathogen. So these antigen-presenting cells can actually bind with the antigen-antibody complex with an Fc receptor. I have the arrow a little bit off, but it should be pointing right here. So after that binding, the APC can then ingest the pathogen and start displaying it to recruit an even larger immune response. Antibody neutralization occurs when the antibodies actually surround a pathogen and thereby block its entry into a cell, preventing an infection by defense, basically. Another way immunoglobulins help the immune response is through binding with this thing called complement. After binding to complement, it can actually assist in the ingestion of the antibody-antigen complex by a phagocytic cell. Or conversely, the complement can come over here and form something called a membrane attack complex, where it drills this hole into the surface of pathogens or even infected human cells. And after drilling this hole, it causes the cell to explode and burst. It's pretty crazy stuff. So here are the different types of immunoglobulins. First, we'll start off with IgM and IgD. These are the first immunoglobulins that appear on lymphocytes in the bone marrow. After traveling through the circulation, they can undergo a class switching. So B cells can class switch from M to D to G to E to A. Now we also have something called secretory IgA over here, also known as SIG-A, and that's a unique dimeric structure that you will find in the gut, and we'll cover that in more detail later. So here we have a table with each antibody and what it responds to, and then the functions. IgM is actually considered a first line of defense, and it also activates complement and aids in the development, maturation, and activation of B cells. Not much is known about IgD, but it is thought to protect against autoimmunity that is caused by IgM. IgG, on the other hand, handles a lot here: mold, bacteria, viruses, parasites. IgG activates CTLs, natural killer cells, macrophages, and complement. It also aids in the destruction of tumor cells. IgE responds to parasites and worms. Unfortunately, it's also well known to trigger allergic diseases. Now, IgA responds to food, vitamin A, and vitamin D. It's also considered the first line of defense. IgA provides mucosal immunity in the gut and bodily secretions. SIG-A has an important role in maintaining the epithelial barrier in the gut, and it makes up 75% of all immunoglobulins. That's why when people say 3/4 of your immune system can be found in the gut, they're referring to this. All right. Here we have a little bit more class switching, and again, depending on the type of cytokine in the environment, it helps to guide what kind of plasma cell the B cell will differentiate into. Now, once the B cell is activated and becomes a plasma cell, the plasma cell is capable of secreting up to 2,000 antibodies per second. So that's it, folks. I hope you enjoyed this fast and furious overview of the immune system. Feel free to leave comments. Feel free to follow along to see future episodes on immunology or other topics. I will be covering a wide range of subjects, including the environment, food policy, science, and just nutrition and wellness in general. So I look forward to continuing the learning experience with you and hope to see you on the next one. Oh, and if you're big on references, I can scroll through my list of references for this episode and the future episodes on the immune system. Hope that helps.